All right. Yeah. So,
So yeah, so you can see the slide, right?
Yeah, yeah. Yes, sir.
Okay.
all right. Yeah. So, so yeah.
Yes, sir, it's visible.
Okay.
Yeah, my voice is going with a little lag. Hello.
Yes, sir, yes.
Okay, fine. So thank you so much for a very nice introduction, and I'm really actually very much honored and very happy that Shrishti has given me an opportunity to discuss with the very enthusiastic young students, and who are definitely the next generation. Even in fact, I'm also very young, so I don't consider myself very old. But anyway, so it's really very very academically pronounced morning, I can say. So I mean, as many of you might have heard that we are working on various aspects of sensor systems, nano engineering and other things. So based on the last about, I mean, as an independent researcher, as an independent PI, so in last 8 9 years, we have worked on various aspects of sensor systems, material engineering, and so many things. So I have meticulously designed this presentation considering the fact, because I know that the participants, they are uh, very budding students, and most of them might be not aware of the very typical terminologies, right? So I mean, I try to tune it to very basic initially, and then I will be moving on to show the detailed improvisation what we can do using the nanomaterials and nanotechnology for a biosensing device fabrication and implementation, right? So that is the basic idea of the whole presentation, what I'm going to have for next about 1 hour. All right, I hope I'm audible, clear, right?
Yes, sir.
Yeah, fine. Thank you. So, uh, so I will be talking about the application and nanotechnology for the biosensing devices. And my slide is also fine, right? You can see my slides.
Yes, sir.
Yes, sir.
Okay, fine. So, so I'm working at Indian Institute of Technology, DHU, and I have an independent lab, and we are working on that. So I will be, discussing about the contents. So these are the content basically I'm going to cover today. What we study at IIT, BHU, Varanasi, and what is nanobiosensors, because whatever we are studying at IIT BHU is basically a sensor system and material engineering. So nanobiosensor is also basically called as a new wave in diagnostics, because I'm sure that everybody who is participating in today's webinar, they might be knowing about many things related to ELISA, and various kind of the molecular biology techniques. So I will try to integrate those things, how, I mean, that idea can be integrated with the sensor system, incorporating various nanomaterials. And another concept is, what can we develop a biosensor without nanomaterials? I mean, is it a mandatory thing to have a nanomaterial in the system? So I will give one or two examples in that also, because the whole idea what Srishi want me to talk today is the incorporation of nanomaterials in the biosensing devices. So I would also talk why we want a nanomaterial. That is a very important point. And after that, I will be discussing the examples of nano engineering system with standard molecules, right? I will be giving few of my work, what we have developed so far. So this is the outline of the whole presentation today. So I'll go one by one on that. So basically, I'll give the brief introduction. As I said, I am working at IIT BHU, Varanasi. So my lab is Laboratory of Biophysical Sensors and Nanobioengineering. So we have diverse kind of things what we are working so far in the laboratory. We can be broadly classified in material engineering and characterization, development of commercially viable nano biosensing prototypes, medical diagnostics, nano engineering, electrochemical biosensors, and paper based sensors, right? So this is the overall area what we are working in the laboratory. Now I will now bring the attention of the students that why we study this. That is very important actually, right? So the basic idea in this case is what, whenever we say a word sensing, so the meaning of sensing immediately go with the process of the detection. You can see the slide also. I am just summarizing the whole slide in this case, right? So when we say detection, it means we are going for a sensing part, right? So I have deliberately put a COVID-19 antigen also. It means anything which is related with a disease, right? Anything which can cause a disease, that can be detected or that can be sensed. That is the basic idea. So these are the few examples. COVID-19 antigen, the whole bacteria, everybody knows that UTI, urinary tract infection is very common, in people. So in that case, the doctor or the pathologist is going to detect a bacterial presence in the urine samples, right? And after that, people can go for the detection of the toxin also, like cholera toxin, vibrio toxin, many toxins. And after that, we can go for the detection of the DNA, RNA, or protein level. If somebody has a bacterial infection, say for example, we can go for a detection of the C-reactive protein or prolactin. So the point is what, we are going to detect a molecule. So that is the basic thing. And that all molecules, they are collectively called as a biomarker, right? So the biomarker can be anything. It can be a whole bacteria, it can be DNA, it can be RNA, it can be a single molecules or anything, right? And when we detect a biomarker, then what we do, we diagnose a disease, right? So that is the main implication of the word detection or sensing. So there are two aspects now. The one of the aspect is detect that antigen or detect the molecule that is present in a diseased person. Like everybody is going for a COVID-19 test, right? They are going to check the antibody presence. Or else you detect the molecule, a chemical or a biological molecule which can cause a disease. For example, mercury, you can detect a mercury also. It can cause a mini matter disease, right? You can detect a lead. You can detect a bacteria in the water. So bacteria has not caused the disease so far, but it has a possibility that bacteria or mercury can cause a disease. So it is not necessary that the molecule what you are going to detect, it should be in your body. It can be in environmental samples also. It can be in the air also, right? So this is the basic idea. And then how to detect these techniques, these molecules? There are many techniques. Everybody know ELISA, HPLC, and so on. So these are very powerful techniques, but the problem in this case is what, they are very laborious. You need a very expert person to do that, and it requires a large handling volume. And if the students who are sitting in the webinar, they might know HPLC, it takes a lot of samples, right, to run the whole column. And it is very time consuming. It's a multi-step process, and you need to have a trained personnel in that. And it doesn't assist the process of a miniaturization. It means at a you have a device, for example, if you can keep it in the pocket, right? And you have a device which you have to house somewhere particularly. So there is a difference. The device which is very small in size, that is called as a miniaturization, that is always required for a personalized diagnosis, right? So the traditional approaches is what, it is not favoring the on-site analysis. You have to go to the laboratory. So in order to remove this complexity, the major problem which occurs in the in the medical diagnosis or environmental monitoring is a quick detection, on-site detection, and ultra sensitive detection. So the new techniques is always required. So these are the basic challenges or the major challenges in the medical diagnosis and healthcare is quick detection, on-site detection, and ultra sensitive detection. And the answer for this particular thing is what, a nanobiosensors, right? A nanobiosensors. Okay. So basically the nanobiosensor, or if you say nanotechnology integrated with the biosensor, right? Because nano can be linked with anywhere. Biosensor is the base, and you can incorporate nanomaterials, then it becomes a nanobiosensor. So nanobiosensor is nothing, it's an analytical device, it's an analytical tool that utilizes a very specific interaction like a lock and key, right? You cannot open the lock with another key. So it incorporates a immunosystem like antigen antibody, a DNA system which will hybridize, because A can't, the adenine cannot interact with the, this one, guanine or cytosine. If you have a stretch of ATGC, then it will be, interacting with the complementary also, or a tissue organelle, or any kind of the receptor system. And then when they have a complexation, then the signal is generated, and the signal could be a electrical, thermal, or optical, or any kind of other signal also. So this particular diagram, if you see, I have very clearly mentioned here the bioreceptor, antibodies, peptides, aptamer, enzyme, or DNA, anything you can use. And these are the molecules which can be integrated with the bioreceptor, and there will be a biological event. Following the biological event, there is a transduction system, and after the transduction system, there will be a signal output. So I'll change the slide now. In this case, you can see very clearly, that these molecules, they have interacted here. There was a bioreceptor, right? It loaded the compound in this particular area, and there was the event, and there is a change in the signal. So what I'm trying to explain here is the bioreceptor and transducer. There are two major things which is present in any biosensor system. And the bioreceptor, as I said earlier also, these are the bioreceptor, and these are the transduction system. Basically, it's a biochemical signal resulting from the interaction of the target molecule, right? So these are the basic components of any biosensor. So as I said, the biosensor is a new wave. So you can see the business market, it is keep on increasing with number of years, and the number of publication is also keep on increasing with number of years. It is very clear that the people are very much interested in academic research, and as well as it has a lot of commercial market. So the basic idea in this case is why people are preferring the nano enabled biosensing system. Why? Because it is very rapid, and you can get a real time monitoring of a disease or any clinical condition. That is very much possible using a nano sensor. The response time is ultra fast. I'll give you the example of a glucometer which is in the market. You just drop a blood, you will get the result out. But in the laboratory also, you can go for a glucose assay, you can go for a carbohydrate analysis, but that is a very traditional way. But that has been improvised using a nano enabled system or integrative devices. So it is very advantageous to have a such kind of system. And another thing is what it go for ultra sensitive or trace analysis. When I say the ultra sensitive or trace analysis, it means the low level analysis. If you are able to detect a single bacteria, like when you, I mean, if people from biotechnology background, they are aware of the CFU, for example, colony forming units of the bacteria. The one unit suggests the population of the bacteria. Now imagine a system, if you can detect a single bacteria, one bacteria, I'm not talking about the CFU, a single bacteria. So that level is also possible, right? Or one compound, it is also possible. All right, so these are the few of the advantages of having a biosensor, which is considered as a wave of diagnostics, right? So these are some market reports. I'll not go in that detail. Right. So the analytical lab, what we see in the near future is something like this. You can see the whole people are working in different places in the laboratory, but every component which has been performed by the individual person can be loaded onto the single chip. The classical example of that is the PCR. So somebody can do on chip PCR also. Somebody can do the separation, the movement, and the detection on a single chip, right? So there are 3-4 different the modules of any assay that can be performed on a single chip. And because of this analysis on a single chip, we called it as a lab on a chip technology, or a lab on a chip system, or a nano biosensing system. So that is the analytical lab, what people see as a futuristic technology, right? Now I'll take a little time on this slide because this is extremely important. So I have mentioned, hello. Okay. So so I have very clearly mentioned, can we develop a biosensor without using a nanomaterial? So the answer is yes. So after this, yes, I am moving to another slide to tell you how a sensor can be developed without a nanomaterial. So this is one of the example what we have done in our laboratory. This is basically a alkaline phosphatase sensor system, right, where no nanomaterial has been utilized, and it worked very well. So this slide shows basically the development of alkaline phosphatase, this molecule, for detection of this molecule in the milk sample. So what has been done in this case, I am talking the fabrication part. So you take a punching machine, you just punch a paper, and the disc will be out. You do some wet chemistry on this surface, and then you anchor a molecule, which is a bioreceptor, what I have shown in the earlier slide. So this part is a probe fabrication. It means this probe is able to detect the molecule either in a standard condition, which is a buffer, or in a milk sample, or in a serum sample, anywhere, right? So so now this will be, subjected for the analysis. So this was made a immune complexation here. Everybody know the antigen antibody reaction. So this immune complexation is occurred, and after that, the inherent ability of this alkaline phosphatase was utilized in order to generate a signal. You can see there is no nanomaterial in this. That's why I have mentioned very clearly no nanomaterial is there. So the inherent property of this molecule towards BCIP was allowed, and then there is a dimeric formation here, and then there is a blue color generation. So overall, what is happening? The white paper turned into a blue color. So the presence of the blue color will clearly indicate the formation of the complexation of the antigen and antibody, right? So this is very, very classical example where no nanomaterial was utilized. And after this process, what happened? It was linked with a mobile phone, and the digital image calorimetry DIC has been performed, and the standard calibration means the range of detection from 1 to 1 million, 1 to 100, whatever, right? So this calibration was performed for the exact analysis. Until this point, it is a yes no. But when it was integrated with the mobile phone, anybody can understand what is the exact level in numerically, the 33.2 or 19.6 or 62.2 or and so on. So the innovation in this case is what it is. It used the waste paper obtained from a punching machine. It is a low cost, affordable technology, and no instrument was required for a sensor development for qualitative assay. Qualitative assay means yes, no, as I said here, and it is very, very robust. It was tested in the real sample, in the blood, or as well as in the in the milk sample, and no toxic chemical and environment. It is very much environmental friendly, and it is a biodegradable system. So these are the advantage of this kind of system. So based on this one, what we did, we developed a prototype. Based on this one. You can see the dimension of this, and this is the final product. What we managed to develop. This is the size of the final device. It is a less than a 5 rupee coin. You can see the final strip. This one, protein detection kit, and the size you can see. You can put it in your wallet also. And the weight of the whole device is 0.38 grams. Right. So this is very much interesting. And the conclusion of this one is it's the first DIC-based protein detection kit, and it merely takes 30 minutes for the whole analysis, and it doesn't require any instrument for a qualitative assay. So what I'm trying to explain with this particular example, that the nanomaterial, it's not must. It is not must to have in a biosensing system. That can also be developed. Now I'll go back to the slide again. So we were here at this point, yes. So what is the answer of this question? Can we develop a biosensor without using nanomaterial? Yes. The example was that what I discussed just now. So now the another thing is what? Then why we are using them? Why we need a nanomaterial in the matrix? Why we need a nanomaterial in the matrix, right? So the answer is basically there are many answers. There are many answers. There are many sub answers. There are many things. But the major thing is what in this case is what the conjugation of the bioreceptor. The second is what the miniaturization and non-enzymatic approach. And the next is the signal amplification. So I have kept few diagrams here which might help you to understand what I'm trying to explain in this case is what the conjugation of the bioreceptor. I'm sure that everybody know about the bioreceptor. It's kind of antibody, what you can see in the slide also. What happens, the antibody is C and N terminal. Everybody know this, right? So the N is NH2 and the C terminal is COOH, carboxylic acid. So what happens if you imagine a system on a particular solid surface, either a glass or any metallic surface, if you drop an antibody and if it works, it's fine. But if you transfer that antibody grafted solid matrix in a blood, right, or in a urine or any matrix where the molecule is there, so there is a very high chance that the antibody might be washed off if it is a very adverse situation. So the antibody should not be washed off. That is why it has been anchored onto the solid matrix. That is called as a conjugation of the receptor system. So the nanomaterial has been greatly, utilized in order to have a bioconjugation matrix, either directly or in a composite form. When I say directly, means the nanomaterial is this, the receptor is this, it makes the complex straight by having a covalent bonding, or else the nanomaterial is there and it has another component, something like a polymer or natural polymers. Hello. Hello.
Yes, sir, we can hear
Yes,
you.
sir.
So can you see the slides, guys?
No, sir, the slides are
No,
not
sir.
visible.
So until what point you have seen the slide? Hello, Dr. Megha.
Conjugation of
Hello, Dr. Megha, are you there? Hello. Guys, where you have seen the slide until what point?
Conjugate.
Conjugation of
Okay. Just wait. Actually, I'm trying to connect with Dr. Megha actually, because unless until she is not making me host, I can't share my slides. Hello, Dr. Megha, are you there? Hello.
Just a minute, sir. There is a small network issue in Srishti organization. They'll be back in 2 minutes.
All right. Thank you. All right. Okay. Hello, guys. Can you see this, right?
yes, sir.
Okay, perfect. So we'll start again. So as I said, there is a conjugation system, right? Either you have a conjugation with a direct nanomaterial, right? Or else you make a composite and then you anchor the receptor molecule. So another concept was what? Another concept was the miniaturization or a non-enzymatic. So you need to have a very tiny kind of a device. So that for that particular reason we use various nanotechnology approaches. And another thing is was a non-enzymatic approach. It means the incorporation of a nanomaterial which can replace the existing enzyme model, right? So that is also called as a nanozyme also sometimes, right? So in this case what is happening, the classical example, you can find so many papers across the globe where people have replaced the glucose oxidase, which is utilized to develop a glucose sensor system, but people have developed various nano structures based on the dendrites or various metallic and non-metallic system, bimetallic, multi-metallic system they have utilized to replace the enzyme. So that is why I'm saying that is one another aspect which people are where people are using the nanomaterials for a non-enzymatic approach. And another is what a signal amplification. This is very, very important actually, because what happens, the signal amplification is directly linked with the concentration basically, right? For example, if you have a molecule, for example 1 millimole, right? For example, at 1 millimole you are getting a signal, right, of any signal. It could be electrical signal or optical signal or a piezoelectric signal or any kind of a signal. But you have done some engineering, nano engineering in that case. At a same, this one, the signal has been improvised somehow. So that is the concept of the signal amplification. I'll discuss with this one in more detail here by this diagram, right? So so in this case what is happening, so I've taken a very classical example that how incorporating nanomaterial will affect the sensor performance. So this is a very, very generalized example. The concentration of blood sugar in the body. One is a hypoglycemia case and one is a hyperglycemia case. See guys, I'm not interested in this whether the people is suffering from a disease. I am interested in the concentration of the sugar in the body, right? So when we say hypoglycemia, we always talk about the level, right? It is 60, 50, that level is there in the body, right? But when we say hyperglycemia, which we say 180, 250, 400, 500, something like that. So that means what we are talking, we are linking the disease with reference to the concentration. So that is why the concentration is extremely important. And if at a low concentration you are getting a signal by incorporating a material, it means it has been obtained because of the signal amplification. At low level you are not getting a signal in a particular system without a material, but when you have incorporated a material in a system, the signal has been improvised, right? So that is extremely important. So based on this idea, the point is basically whenever we go for the analysis of any sensor, there is a very standard plot. And this I have incorporated the standard plot. See why I'm talking this, because this is always present in any sensor, with nanomaterial, without nanomaterial, various type of sensor transduction system, either electrochemical or optical or piezoelectric or colorimetric, anything. So this graph is always common. This graph is always common. So that is why I will discuss this one, so that when I'm giving the real example of the cases, what people have done, so you will understand what is actually I'm talking in this case. So whenever people are developing any sensor, they are very much interested in a dynamic range. It means the range in the range where when you are increasing the concentration the signal when the signal is keep on increasing, right? So that is very, very much important. It should be as wide as possible. Now you see this graph here, the black one. If you have made a sensor which is going very well, which is very powerful, but it is a very narrow range it is going to act. For example, it is measuring the glucose from 100 to 150, for example. So the possibility to detect the glucose in a hypoglycemic condition or a hyperglycemic condition is zero. So that is why we are always aiming to have a sensor which has a wide dynamic range from, for example, specifically for a glucose case, for example, 30 to 500. If it is able to detect glucose from 30 to 500, then the range is very wide. And then whatever is the situation, it is a normal glucose level or a hypoglycemic condition or a hyperglycemic condition. In every case, it is going to work very well, right? So this is very much important. And these are other terminologies. For example, LOQ or LOD. LOD is a limit of quantification. LOD is a limit of detection, which is the analytical performance. These are the analytical performance values. And LOL is basically limit of linearity. So it means the maximum limit at which the sensor is able to detect the molecules, right? For example, if I say in a general word, if you have, for example, 100 chairs, only 100 people can fit on that. So it means there is a saturation level. Same with the sensor system. If you have antibodies 100 loaded onto the chip, only 100 antigen is going to bind. Over 100, it will be saturated curve. So that's what it is called as a LOL. It's a limit of linearity, right? So again, the nanomaterial and biosensor, what is the take home message from this particular discussion is what? It improves the detection range, it stabilizes the system, provides a robust system, assists in biocompatibility as well, bioreceptor immobilization, I have given the examples, more sensitivity, improved signal to noise ratio, and overall performance of any sensor is improved greatly when you are incorporating various nanomaterials during the sensor fabrication process. So this is one of the, the whole layout, what I'm trying to explain here. From the layer 1 to layer final layer, or there are various, approaches. We can improve the signals, the increase in the conductivity of the electrode, if it is an electron transfer system, direct connection of the redox side to the electrode, and so on. There are many things. And now you can see from this is one of the electrochemical readout system called as a cyclic voltammetry. So the important thing what you have to note is basically this curve, black one is without nanomaterial, right? But when you have incorporated the nanomaterial, you can see the signal has been improvised. The signal is enhanced. So that is what I'm trying to, hit in your mind, that whenever you are incorporating a material, the signal, it is improvised. It is the higher signal you're getting. And in addition to this one, what all I have listed here is also very much important in case of the nanomaterials and biosensors. So what is so special about the nanomaterials? It has a lot of unique features such as optical, electrical, mechanical, magnetic, and thermal properties. You can tune it. It is highly tunable system. So by tuning the system, by tuning the nanomaterial, you can change the performance of the sensor. That's what is very much important when we integrate any nanomaterial in any kind of the sensor system, right? So these are the basic classification. You can find some other classification also, but I've taken this classification from a standard book, right? So the nanomaterials, it could be a nano in form of particle, carbon-based nanomaterials, dendrites, quantum dots, conjugates. Conjugate means there are two nanomaterials, they are coming together. It could be two particle also. It could be a polymeric system or a carbon-based material. Graphene is very much common, carbon nanotube. And a nanoparticle, it could be a metallic nanoparticle, it could be a polymeric nanoparticle, it could be a metal oxide nanoparticle. So these are the general classification of the various nanomaterials. So before going in more detail about the incorporation of the nanomaterial in the sensing system, there are a few terminologies more you have to understand, or the approach. I mean, whenever you are going to study the nanomaterial integrated sensor system, what you have to know, you have to know what to detect. I mean, if you don't know what to detect, you cannot make a sensor, right? And after this process of what to detect, you can go for the scheme generation. The scheme means the design. I mean, how you are going to approach with the whole process, right? You want to use a carbon nanotube, you want to use an aptamer in a system, you want to use an enzyme, or you want to use what? So the selection of the material, the selection of the receptor, the fabrication approach, it largely depends what you are going to detect. After this process, you will fabricate. Physically, you will be doing that you will use various techniques. I will not go in detail what techniques you will be using, but you will be fabricating. Physically, you will be doing that. And after that fabrication process, when we say that we have done it, so you need to confirm it, that whether what you have done is correct or not. So that correctness of the system is called as a characterization. There are various methods to characterize. I'll show in the next slide. After the characterization process, you will be going for the optimization, that in this optimum situation the maximum signal will be appearing. You will obtain a maximum signal in this particular optimization system. So after this optimization, we'll moving for the analytical performance. It means The graph what I discussed in the previous slide, LOD, LOQ, LOL, and dynamic range. So these things you will be calculating, you will be analyzing these parameters. In addition to these parameters, there are many more things what we will be analyzing. That all comes in the analytical performance values. And the next step is what? When you tested the analytical performance in a standard case, after this process you will be challenging the system in a real sample model. For example, you have, if you have developed a glucose sensor, so you will be eventually testing it either in a blood, either in a serum, either in a juice, or either in a any matrix wherever the glucose is actually present. For example, if we have developed a analytical sensor for detection of a cancer biomarker, for example, HER2, it's a breast cancer biomarker. You will not test that HER2 cancer biomarker or a cell, SKBR3 cell, breast cancer cell. You will not be testing that cell or the protein in soil. It doesn't make sense. So that real sample matrix is very much important to design, to select. That is why we do the real sample analysis, and then we need to check the interference of the molecules. If you made a glucose sensor, but it is also able to detect fructose, it is also able to detect other monosaccharides or disaccharides. So there is no point. The point is basically it should be extremely, selective. If you are making a sensor for a breast cancer biomarker, it should not go for any cervical cancer or any blood cancer. Otherwise, there is a cross reactivity, there will be false positive results. So that is what is called as the interference analysis or the selectivity assay. And the final is the stability. Stability is very, very much important when you go for the commercialization. If you are housing it somewhere in Chennai, if you are to transfer it to Delhi or Assam, somewhere else, the system should be very stable, right? The shelf life, it should be very much high. So these are the the general information whenever we are actually doing the designing of the sensor, right? Okay, so these are the sensing modules what I'm going to talk now. So these are the sensing modules what we have developed recently in our lab. So there is one more area what we work, but I will not talk that. It's a nanomedicine, but I'll be talking about various other modules what I'll show in the next few slides. These are areas basically: the paper microfluidics, the cancer biomarker based sensor, the in vivo sensor we have developed so far, ex vivo actually, for the ascorbic acid detection. And these are the various nanomaterials what we have recently developed. There are few actually, we have developed a lot more. So for example, the bimetallic system, the nano rattles, the bimetallic gold and iron nanoparticle dendrites and nano switch, and so on. There are much more things what I'll be talking in later slides. So the nano bioengineered sensing system developed at my laboratory, I'll be giving now examples. So I have discussed so far about the receptor, about the nanomaterial, what can be used, why we are using a nanomaterial. I gave example. There are three things, as I said earlier also. So these things you will be looking directly in the example what I'm going to talk now. So before going into that, see, the sensor can be of various types. Whatever I have discussed so far in case of the ALP1, that's a optical sensor. There is no, it was not an electrochemical sensor. So I'll talk now onwards. I will talking about electrochemical nanobiosensors, where the various nanostructure will be there. So before going into the in the detail of this system, it's basically a redox reaction. So whenever we are going for an electrochemical biosensor, we go for the analysis of the redox system. For example, copper goes to copper 0. So there is a reduction potential, and when zinc Zn go to Z plus 2, there is oxidation potential. It means in general I am trying to explain, at this potential this reaction will be happening. And when we go for a electrochemical nanobiosensor, we use a 3 electrode system. One is a reference electrode, one is a working electrode which is called as a biosensor, one is a counter electrode. And this is the basic principle. There is a substrate, and onto the chip, the solid matrix, we will load all the things, either enzyme or the nanomaterials. When you apply a voltage, for example this one, and then the product will be out, you will get a signal. So this is the classical output of electrochemical signal, which is called as a cyclic voltammetry. So if I read out the electrochemical nanobiosensor system, it's a self-contained integrated device, right, which is capable of providing a very specialized qualitative or a semi-quantitative of highly precise signal output. And it only happens when the biorecognition element is retained in the direct spatial contact with the transduction element. So that is very, very much important. So now onwards, whatever I'll be talking where the readout is the current, right? So this is very much important when we, I mean, after this slide, whatever I'm talking, it will be for a readout system where we will be monitoring the current values, right? That's why I deliberately kept this slide here. So before going again further, these are the basic, information for the nanomaterial characterization. There are many more. So these are the steps. Whenever we go for a nanomaterial research, in case of the biosensing, we go for the synthesis process first. We need to synthesize, and then we go for the characterization. What I have mentioned, it could be XRD, SAD, EDX, FTIR, and so on. There are many things. So this is the general information of the nanomaterial characterization, and these are the analytical procedures and the terminologies. So whenever we develop a sensor comprising a nanomaterial, that is called as a pro-fabrication, and after this process we will go for the characterization. The characterization means whether the sensor has been developed correctly or not, incorporating the bioreceptor and nanomaterial. So we will be optimizing the pH optimization, we'll go for a temperature optimization, we'll go for the electrode characterization, where we'll study the electrode, the scan rate, and so on. In case of the analysis part, we will be studying about the response study, the selectivity coefficients, and so many things. So after this whole exercise, what we will get the desired sensor. That's the final product of that whole research, right? So these are the few things what I'll talk now. This already I explained. So this is one of the advanced version of the cyclic voltammetry. This is called the differential pulse voltammetry. It's a potential dynamic technique. You can see the potential is sweeping on the x-axis and the current is getting out of this. So I'll give you one example where the surface is very much conducting, but when you put a protein onto the surface, a metallic surface, and you measure the current across this one, so protein, when you are loading, it will increase the insulation on the surface. It means the resistance will be increasing. So that is also measured by electrochemical impedance spectroscopy, right? So you can see very clearly, this is the impedance values. The resistance is on the x-axis. I'll not talk why this is z hyphen or this one. So this part, the resistance is increasing. This is the normal the antibody, and when the antigen is binding, the resistance is increasing. That's what I said. So this will also come somewhere when I discuss the result part, because when you are talking the incorporation of the nanomaterial, we need to discuss the whole idea in terms of the results. Then you will understand why the nanomaterial is there sitting in the sensor design. All right, so I'll now talk on the typical examples where a lot type of nanomaterial has been utilized to develop various kind of the sensors. So I've categorized a very recent type of the nano system. I'm sure that many of the students, they have heard about the nanoparticles, gold, silver, and so on. But these are the some unique type of the structures. That's called as a nanodendrites, metallic nanodendrites. It's a branch three-dimensional structures. So I've taken the example of electrocatalysis of the gold nanodendrites, and after that I've taken the example of a nano hybrid system where the nano tubes, nanoparticles, and the dendrites, they are all together will be sitting on the sensor surface, right? And after this process, I'll be talking about some other bimetallic system, and then various other immunosensors. So the idea in this whole case is what? From a small molecule, from a free radical, up to the protein molecule. You can imagine the hydrogen peroxide to a final protein. So that's why I have taken this range of molecule, right? So it is always possible to detect from smallest to the toughest. So that's what I've taken this particular example. So I'll talk a little about the dendritic structures. So dendritic structure is basically the branch structures. So I'm sure that everybody know what is dendrites in the nervous system. So this type of structures, they are present in in normal things, the silver fern or snow formation, if you break the ice there. So this kind of structure you can see naturally. So this kind of structure can be engineered. These nano structures can be engineered onto the chip, and which is having a central branch followed by a second 2-degree branch and the third degree branch. So this is very much possible by having an electrochemical nano tuning concept. So that what we also develop. So this is a metallic dendrite. You can see it's a tree shaped structure. What I have shown in the previous slide, this one, this fern. So this similar pattern can be obtained onto the sensor surface also. So the advantage of this system is what? It's high density electroactive sites you will get. You will getting the higher the area. So, and this structure is highly tunable. If you want to do the nano engineering part in order to change the signal parameters, you can do that also. So that's what very much important in case of the nano dendrites. Now the mechanism how it forms is basically what, there is a precursor molecule. You can see from A to D, there is a precursor molecule, and after this process, there is a nucleation process, and after that process, there is an aggregation process, which will lead to the primary branch formation, and followed by, when you will do the process of the reduction, keep on doing that. So there will be formation of primary structure, secondary and tertiary on this particular chip surface. So that is a highly tunable system. I'll not go in that much detail how it works, how it forms, but eventually there is a three-dimensional structure which will be formed onto the surface, which is very much possible. So based on this idea, I'll be talking about the nanodendritic nano chip for a label-free hydrogen peroxide detection in the blood sample. So this is basically a very classical example where a waste to value based concept has been utilized in my laboratory, and we have developed a nanodendritic chip on a ITO glass, that's indium tin oxide glass surface. So now you can see what I said, the nano tuning. You can see there is a deposition potential. I'll not talk. This is a basically reduction process we are doing in this case. So when we increase the time of reduction, you can see the formation of the structures has been changed. You can see it is keep on forming. And when we go for a 1500 second, the typical fern-like structure it has been formed on the surface. So these are same micrograph of gold nanodendrites, which has been used, which has been developed after the process of electrode deposition. So after this process, what happened, as I said earlier also, the signal was increased. Here the current is increasing on the y-axis, you can see here. And when we measured the resistance, what I discussed previously also about the insulation process, so the resistance is less. It means the conductivity is higher. So this is one of the data I'll not talk. This is more, very, different kind of data that's not required. But this shows basically the stability of the system, right? So this is what I want to convey here. So after this process, as I said, it's a three-dimensional structure. You can see this is the horizontal cross section view. You can see, right? It grows like like a 3D structure onto the surface. That's basically basically a three-dimensional nanodendrite. You can see very clear images here. So after this process, we have done the whole characterization, and these are the values which shows the diffusion coefficient values, how the molecule is diffusing to the electrode surface. And eventually after this process, we moved on to the detection of the hydrogen peroxide using the biosensing system, right? What we have developed, right? So in this case, what we have taken the hydrogen peroxide because of that it has a lot of biological importance in the system. And then this is the final fabrication process, you can see. So this is the electrode deposition, as I have discussed earlier, using the gold system. And we have done a sputtering process, right? That's a very very known process when you go for the SEM analysis. So this has improvised the system, and then hydrogen peroxide has been detected by this particular reaction directly in the blood sample. So these are the data what I will not discuss in more detail. So basically, this is the step-by-step characterization of the system. And when you are increasing the concentration of hydrogen peroxide, the signal is keep on increasing. What is most important to discuss here is what, when we have injected the hydrogen peroxide, you can see the response time is less than a second. Within 1 second, you are able to detect hydrogen peroxide very accurately. So that is the importance of incorporation of nanomaterial in the sensing module. Otherwise, if there was no nanomaterial, no nanodendrite, we were not getting any signal, right? So now this is the final output of the hydrogen peroxide based sensing system. You increase the concentration from 10 to the power -5 to 10 to the power -12. The signal is keep on increasing, right? And then we made a standard calibration plot, what I discussed earlier also with LOD, LOQ, and LOL and dynamic range. This is that plot. That's why I discussed that plot separately. So these are the values. Like LOD could be was a 9.8 10 to the power -13 moles. This is very, very low, right? So these are the interference studies where you can see that it is highly selective. What I already discussed, the selectivity is very much important. So these molecules, they coexist with hydrogen peroxide, but they doesn't interfere with the detection of the hydrogen peroxide. All right. So we have also checked the market potential of the sensor. You can see in the clinical concentration where it what is the range of the hydrogen peroxide, 10 to the minus 6 to 10 to the minus 9. And FDA approved maximum concentration of hydrogen peroxide milk is 10 to the minus 6. And we were able to detect from minus 5 to minus 12, right? So this is very much important when you are going for the commercialization of the sensor system, right? So this was one aspect. So this is the comparison of the sensor what we developed. This was the research highlights. We have developed this system, right? So these are significance, what I already discussed. So I'll be moving on to another type of the system. So I'll talking in this case the bimetallic system. Until now we have talked a monometallic nanodendrite. It's a bimetallic nanodendrite. So I'll skip the known things what I have already discussed. I'll be talking the incorporation of these two metal in a one system, right? So yeah, so this part I have deliberately circled here. So for example, this is the chip, right? This is the black part. So when gold is separately developed, you're getting a signal. When the copper is separately developed, you're getting a signal, but this is very high. The maximum signal, the green curve you can see here. And similarly, complementary experiment has been done because we are monitoring the current response here, the conductivity of the system. But in this case, what is happening, we are going to check the resistance of the system. The highest conductivity means the lowest resistance. You can see from this histogram also. So that is why this is the importance, guys. This is the importance of the system, where the nanomaterials, which is a bimetallic system, when the bimetallic was loaded onto the chip, the signal is extremely high compared to the individual monometallic state of the system what I have shown here. So this is the most important result of this part, right? So this is again the characterization using the SEM images and the optimization at how much time we will able to do that. This is the what I said. It's a monometallic means. This is the basic the mapping result. You can see gold, the presence of gold and copper both in the system, right? And this is the dark field TEM images. This is the uh, normal TEM image, but when we have done the mapping, this is the copper and this is the gold, which is incorporated in the position of the analysis is same basically. Now you can see this is the normal situation, but when we tested the presence of copper, when we have tested the presence of gold, they are separately present. But when we mapped it together, when we have merged it, then you can see gold and copper is merged, and it is very much clear that both the type of metal nanostructure is present in the matrix. So after this process, what has been done? Again, the highest signal, gold and copper is they are giving the highest signal compared to other one. And after this process, what happened? We make a standard plot. Again, that standard calibration plot, what I discussed earlier also, LOD, LOP, LOL, and the dynamic range. And based on this plot, you can get the final results, something like this. So here are also from 100 to 1000, when you are increasing the concentration, you are getting a straight line. It means it works very well. The meaning is this, and the LOD in this case is what 8.05 nanomoles. Again, this is the interference analysis and the real sample analysis using the urine sample. So you can see the standard buffer, the red plot, and the urine sample, the black plot. It works very well. The sensor works very well in both the cases. So again, this is the one of the more example where the nanohybrid system has been used. The nanotubes, the nanoparticles. This is the nanohybrid, and onto this nanohybrid, the dendrite has been grown. So what we discussed so far is first was the monodendrite, second is what gold and copper, and third is what onto the nanohybrid the gold is going to be formed. So again, in this case, what you can see, this is the hybrid formation, the nanohybrid formation, the nanoparticles and the nanotubes. And after this process, what you can see, this is a very beautiful image. You can see, so this is the particle area, and this is the nanotubes, and onto this we have grown the nanodendrites like this. And this was also confirmed by the mapping also, right? So this part is basically the dendrite. These are the tubes, very clear, and these are the dot are the particles. So on this one system, there are three type of nanomaterials which will assist in the formation of the final sensor. One nanomaterial is a gold nanoparticles, another nanomaterial is a nanotubes, and third is a nanodendrite. That is very much important, right? And this is very classical study where different nanostructures has been utilized. Again, we have compared the performance parameters where the incorporation of the various, as I said, the 3 nanoparticles, the nanomaterial has been incorporated, the signal is maximum, and which was also complemented by the impedance spectroscopy studies. So again, in this case, you can see this is the normal electrode, the normal chip where there was no nanomaterials. After the incorporation of the nanomaterial, the signal is completely jumped. What I discussed, if you remember earlier, the signal amplification. That's what is the example, actual real example of our the database example actually in this case. Again, when uh, the signal was increasing when the concentration of the target was increasing. In this case, acetaminophen was the target, and you are getting the straight line with this one. And this is the again the final result when the concentration of acetaminophen was increasing, keep on increasing. The signal is keep on increasing from between 100 to 750 nanomoles, and this is the straight line. So in this case, the response time is 10 seconds. Right. So this is the importance of nanomaterials. What we are discussing with reference to the biosensor development. So again, the interference in the real sample analysis has been tested in the urine sample, and it works very well, as you can see from this plot. So this is the chip to chip variation. This is also I have kept in the slides, because when you are testing the chip, you have to check with multiple chips, because maybe chip number 1 is working well, chip number 2 is not working well. So you have to take a n number of chips to confirm the final output. So this slide shows the 5 different chips have been tested, which was separately designed. And after this process also, the analysis part, the signal is more or less same. So it means it works very well. And from week 1 to week 8, there was no problem in the signal. It means it is stable up to almost 2 months. So this is also the comparison of the various data what has been developed for the acetaminophen sensor. And when you have incorporated various different type of nanomaterial in the matrix, the performance is definitely very high. Now, what I discussed so far, so I discussed very small molecules. So dendritic system or a dendroid system can also be incorporated for the development of the bigger molecules as a target. In this case, the molecule is again the ALP, alkaline phosphatase. So in this case, what happened? This is the bare chip, right? On this bare chip, a gold nanoparticle has been incorporated, and after this gold nanoparticle incorporation, the nanodendroid has been loaded onto the chip. That there are 2 nanomaterials. After this process, what happened? There is a addition of the functional groups, carboxylic acid group, which is coming from because of the graphene oxide, which is also a nanomaterial, 2D nanomaterial. And after this process, what happened? The antibody has been anchored, as I said also. The nanomaterial assisted anchoring of the bioreceptor is the example here, basically what I discussed earlier also. So this whole complexation is assisted through the incorporation of the nanomaterial in the matrix. And then this whole probe was exposed towards the detection of the ALP in the clinical serum sample. This is another type of the example where nanostructure has been utilized to develop the immunosensor in this case, right? And then again, as I said, when you are loading the ALP in this case, the insulation is keep on increasing and the resistance will be increasing. So these are the prospects basically. The receptor can be loaded for other molecules. In this case, the multiplexing is possible. It means in one go, you can go for the 4 or 5 molecules in one shot. And it takes nearly 15 minutes to detect the molecule. And basically, when we go for the analysis by using this technique, it's label free. Like if you go for the ELISA, you have to add some labeling molecules, but in this case you don't need to label this. And this can also be integrated with the smartphone. So that is also one of the prospects of this kind of the nano sensor system. So this is the last example what I'm going to talk, where until now we have incorporated the nanomaterial which assist in the formation of the signal. But this is one very classical example where a non-invasive oral cancer sensor has been developed by electrochemically active nanomaterial, right? So the importance of this particular example is what? The material itself is giving a signal. Unlike other methods, you have to add something in the buffer or somewhere to obtain the signal. But in this case, there is no need, absolutely. So I'll talk a little here in this scheme. the nanomaterial has to be synthesized. It's a microwave assisted method. I'll not go in that detail. So the nanomaterial which has been developed here is gadolinium hexacyanophyrate. So this material is electrochemically active. When I say electrochemically active, it means it gives a redox signal. When it gives a redox signal, it means it is you can get a classical curve of a cyclic voltammogram, what I've shown earlier also, and in this diagram also you can see here. So now the point is, until this point, you, if the sensor surface is having this material, you will get a signal. But when you are loading the material which is insulating in nature, there will be a drop in the signal. So that is the basis in this case where this type of nanomaterial has been utilized to develop the oral cancer sensor. So I'll talk a little here. So this is the characterization of the material. That's not that important. So this is also one of the characterization of the material, the TEM analysis and SAD pattern. So this is the important thing what I want to discuss here. So this is the blank signal. You can see in the blank also you are getting the signal. And when you are keep on increasing the concentration of the oral cancer marker, the signal is dropping. Earlier you have seen the signal was increasing in positive correlation. But in this case, when you are adding the target molecule, that's the example of CIFRA 21, it's the oral cancer marker. So when you're adding this one, the signal is keep on decreasing because of the increase in the insulation, right? So something here, I put this diagram here. Hello. Yeah. So I've kept this diagram here. So until green one, you are getting a black curve, but when you are increasing the CIFRA 21 molecule here, this complexation is increasing the insulation. There is a drop in the signal, the current one. So this is one of the example of the self-reporting sensor based on the electrochemically active nanostructures. So the deployability of the sensor is what? Again, you have to check the interference with other molecules. This is one of the oral cancer marker. This is a protein basically. So you have to test with other proteins. In addition to other proteins, you have to check with the other cancer biomarkers and other molecules which is present in the real sample matrix. And again, in this case, that has to be checked in saliva also, because the saliva contains this molecule when there is an oral cancer case. That is why I said the selection of the real sample matrix is very much important, right? Okay. So this is the significance of such kind of the nano-based sensor system. It's very wide dynamic range. It is in the clinical range, basically from 2 to 50 nanogram per ml. It's a clinically relevant range, and the LOD is 0.6 nanogram per ml, right? So this is the end of the significance of this such kind of thing. So I'll be concluding the whole talk now. So overall conclusion and the vision is what? Basically the disease or any situation where the molecule has to be tracked, that can be done in a personalized setting. That is very much possible. And the information of the novel biomarker with high specificity and their level is very much important. So I said in the last slide deliberately 2 to 50, because if the sensor is not able to detect between this range, the sensor has no meaning, because that cannot be deployed for point of care analysis or for clinical examination. So the range where the molecule is present either in the environment or in a real matrix, urine, blood or serum, whatever, that is very much important. You have to take into consideration when you're designing a nano sensor. And again, the new nanomaterials can be engineered for diverse sensing applications. Okay. And the multiplexing system is also very much upcoming things, because in one shot you can go for at least 5, 10, 15. People have done that also. People are doing nowadays also. So that is very much important, and that can be done by incorporating various nanomaterials, and which eventually lead for the integrated devices. Right. So this is the overall conclusion, and the conclusion with respect to the biosensor, you need to have a knowledge of various backgrounds: biochemistry, instrumentation, the basic biology, clinical science, chemistry, nano engineering, and so on. And most important, you need to know the market requirement. What is the market requirement? If the market doesn't require the sensor X, you no need to develop that sensor X, right? Because that's basically the applied research. So you need to apply that for the community basically. And what information the sensor is going to give you? It's analytical information. The sensor gives you the values. I said in the initial discussion also, we are interested in the levels. I mentioned this in the slide, one of the slide. The sensor output is the level, is the concentration, the selectivity process. So based on this data, the healthcare professional or the environmental expert, they will give their recommendations. If the doctor knows the level of the glucose is this, he will tell you whether it is a hypoglycemia or hyperglycemia or somebody is normal. If the water contains mercury, the environmental experts will say that what water is not potable, the bacterial load is high. So the values, the numerical values are a semi-quantitative, semi-qualitative. This kind of the values are very much important when we talk with reference to sensor design. So guys, thank you very much. So I'd like to acknowledge my whole team of people who work with me and who are working with me, and the funding agency SARB, DBT, IIT BHU, and IIT Guwahati also, where I work. So thank you very much once again. So these are my contacts detail. If everybody has some question after this presentation also, they can write me. And thank you so much.
Thank you, sir. Students, so now you can ask and discuss your queries with Professor.
Good afternoon, sir. I have a question on gold nanoparticle related biosensors. So when we take biosensors, we often end up with an aptamer protein or an antibody. But some kinds, for some instances, we have proteins that are very costly. For example, we get only 10 micrograms or 25,000.
Right.
So, but then they might be the only reliable way for detecting a particular disease through a sensing system. Others might be further costly. So I hope nanobiosensors would be the most economical way to achieve that because it will use very less amount. One challenge that we face is when we conjugate that kind of a protein to a gold nanoparticle, let us take gold spheres for example,
Right.
there is an issue of curvature. We tend to have a very small quantity, nanograms of protein, so it generates very less amount of signal for electrochemical detection.
Right.
So we shift for optical detection because the gold has this plasmon resonance
Right.
principle.
Sure.
So when we conjugate a protein at 20 nanometer spheres, the curvature is higher and there is less chances of protein getting absorbed even by gold thiol chemistry. So if we go for higher nanometers, the color intensity is less. So relatively less than the 20 nanometer gold. So
Okay.
how can we overcome this issue?
See, I understood your point. First thing is, I'll answer you step by step. So you said that you need to have either aptamer or antibody, right? In this case, when you are designing a sensor, right? See, it is not mandatory. So I have shown one slide where I mentioned start studying the sensors. Right? I mentioned the first box was what to detect. If you are aiming the detection of any protein or any protein biomarker or something like that, so you need to have a receptor molecule, because that molecule is not going to to give a signal, right? So to make the sensor selective, you have to have a receptor, otherwise it will not work. First thing. Another thing, if you're going for the chemical based molecules, right? For example, acetaminophen or hydrogen peroxide or something like that. In that case, it is not necessary to have a bioreceptor like antibody or aptamer, which you said it is expensive, but we cannot, we cannot avoid that because that has to be there in the system. Even for COVID-19, you can see people are going for the antibody based assay. So again, so that molecule which is, for example, electrochemically active or optically active, so that can be directly related. So another issue with the direct electron transfer based sensor is what? The overlapping of the potential. If you are working in that area, you might be knowing this. So sometimes at a similar potential, you are getting the signal of various other compounds also, which is the interfering molecules. So in this case, that's a play of the nanomaterials. So you have to engineer the material in a way so that the generation of the signal should be for that particular molecule against which you are making a sensor, right? So that is very much important. That's why I said last week also the engineering of the nanomaterials, right? So that's what I said. The antibody based sensor is different, the nano material incorporated sensor is different. For example, glucose also, glucose, everybody using glucose oxidase, but you can use a dendrite also. Even many people have done, we have also done that. without nanomaterial, without enzyme, you can go for a glucose analysis. You understand what I said?
Yes, sir.
So you understood I mean what I explained to you?
Yes, sir. Yes, sir. It is clear.
Is the answer what you were looking for?
Yes, sir. It is almost near, but I have a still I have a question on how to overcome the challenges on this part, that is attaching a protein to a less curvature nanomaterial. So
See,
I
I
think that
you
from
are
your
talking
answer,
with reference to the size, right?
sir.
You are talking with reference to the size of the particle, nanoparticle,
Yes,
right?
sir.
See, you can do one thing. I mean, in my experience, what is the size you are handling? 20 nanometers or less than that?
So the nanoparticles I'm using are in 20 nanometer, 10 nanometer, and 40.
Okay, so see, 20 I know very well. In 20 people have done that, because I know, because you are talking when you are loading a high concentration of antibody, the problem in this case is what? There is steric hindrance, right?
Yes, sir.
Many of the because it's a bulky molecule, right?
Yes,
So
sir.
that is why if you read some papers, I'm sure maybe you are doing a self assembly or something like that, maybe,
Yes, sir.
right? So please don't do that. You can do one more thing. You can put a capping agent, something which has a flanking group. If you read a paper from Chat's American group, right, they are utilizing a lot of other linkers in between. So might
Mm.
be incorporating that linkers, the loading of the antibody or this one, the aptamer might be, it would be fine. But definitely the SAM using the antibody is not that recommendable, because I mean, if you're particularly when you're going for optical detection, because it might lose the binding, and especially in case of the real sample analysis in the blood or in the serum or in urine. So that makes a problem. So my suggestion would be, you can add a linker in between and try it once more.
Yes, sir. Sure,
Because
sir.
20 to 30 is fine. I know that 20 to 30 nanometers people have done a lot loading of the antibody in an aptamer and other receptors, right?
Yes, sir.
Okay. All right.
Sure, sir. Thank you, sir.
Yeah, no problem.
Any question now? So if you can't explain, you can write down in the chat. That will be also fine. Actually, you should utilize this opportunity interaction with Dr. Pranjal. It's a good opportunity to ask and discuss your queries. Did you understand the whole presentation?
Yes, ma'am. Sir, another question I have is
Yeah.
uh, while while working on a project, actually it is my mentor's project. While working on it, we faced a challenge in detecting hydrogen ions from a serum sample because of protein cross interactions. But then that actually led us to lose some signal, and it gave us variable results.
Right.
So we have a challenge to detect hydrogen ions alone from a
Mhm.
heterogeneous sample that has different proteins
What
capable of
you
cross
detected
interactions.
hydrazine or hydrogen?
Hydrogen, H plus
Hydrogen.
ions.
Okay. All right.
Uh,
All right. All right.
so the proteins usually make us lose the signals of cross interaction. Is there any chance of a nano hybrid that can potentially deal with this problem?
So, I mean, what I understood, your target is hydrogen ion, right?
Yes, sir. That is our signal.
And what is the matrix where you use this?
It was a
Your
carbon nano matrix, sir.
no, no, no. the matrix I'm talking, you said that when you are testing in a real sample, then it is
Yes,
making a
sir.
problem, right? What is the real
Yes, sir.
sample you are using?
Serum.
Okay, so right, so I mean that you can try one thing. In my opinion, you can go for a standard addition method, right? So you take a serum, or you can find some of my papers. We have mentioned very clearly that. So you can take a standard addition method, which standard addition method says you take a serum sample and you just dilute it for 10 times, 20 times. You have to do that 5, 10, 20. You have to do the experiment for each and every point, and then
Yes,
you
sir.
do the analysis. First time you test without adding the hydrogen peroxide, say this one hydrogen, you detect for hydrogen ions
Yes,
without,
sir.
I mean, the adding. So if you're getting a signal, then you keep on adding that. So that is why I'm saying this is a standard addition method. You add and then you make a plot, straight plot. After this, you will get a, the values on the x-axis something. So I mean, I don't have that plot, otherwise I could have shown you. I mean, conclusively, what I will advise, you go for a standard addition plot. So the factor of which you have diluted the sample, for example, in the diluted sample you are getting, for example, 10 mg per ml, for example, for
Yes,
any
sir.
standard molecule, you just multiply 10 mg per ml for that factor of which you have diluted the sample. If 10 times diluted, you multiply by 10 factor, so you will know the actual concentration of the molecule in the original matrix, right?
Okay.
So that's
Yes,
how
sir.
it works. So the bottom line of the whole discussion with me with reference to this hydrogen ion is what? Standard addition method. Just try this.
Okay, sir.
You can
I'll
find
definitely try.
this method in our papers. We have done it so many times. The
Okay, sir.
protocol is there actually.
Yes, sir. Sure,
All
sir.
right. Yeah. All the best.
Thank you, sir.
Yeah. No problem.
Sir, how we select which nanoparticles? Okay, sir, there is a question in chat box.
Mm-hmm.
How we select which nanoparticle is suitable for immobilizing in the bioreceptor for detection?
Okay. See, generally what happens, if you see, there are so many papers on gold nanoparticles. Yeah, there are so many papers on silver. I mean, there are, as I said, it depends upon the application, right? For example, if you're going for a electronic sensor, right? So generally we go for a metallic nanoparticles, right? For example, as I discussed with Yogesh also, I guess. So gold, if you are selecting, for example, the immobilization of any receptor or any, for example, cysteine kind of a molecule which has a thiol group. So you will be using a gold nanoparticle because of the ability to for self-assembly, right? And moreover, you will not select a metal oxide based nanoparticle, for example, zinc oxide or any other oxide based nanoparticle, because that is not that conducting compared to the metallic nanoparticles, for example, the gold or the silver or platinum. So again, as I said, it depends on the what type of the transduction system is there, and whether you are able to utilize that nanoparticle for further engineering it in order to anchor the molecule, right? So that is the overall idea of selection of the nanoparticles. And moreover, there are reports which where the people explore the possibility of the nanoparticles which has not been explored. So that is also one of the area what people are working, that this nanoparticle or this nanoparticle composite has not been tried. Let us try and let us check how it works in a sensor design. So that is also one of the aspects people are working on that. Okay.
Hello, sir.
Yeah, please.
Good afternoon. I'm Satyavat. Sir, actually, we know that a crowded environment exists inside a cell structure,
Okay.
in which the cytoplasm is typically different from the dilute solutions,
Right.
generally used in vital of proteins.
Right.
So my question is there any significantly affect of that behavior on the proteins? Does it affect significantly the behavior of proteins?
No, I don't understand. I mean, you are talking about the protein which is within the cell.
Yes, sir. Within the cell, sir.
No, I don't understand. I mean, is it related with the sensor or what? This question. I mean, if for example,
Yes,
if
sir.
some,
Yes, sir.
okay,
Related to sensors, sir.
how it is related? Can you? I'm not able to understand what you're asking.
Sir, actually, my point is that inside a cell, there exists a crowded environment in a cell structure, right,
Okay,
sir?
right,
In which cytoplasm is typically different from the dilute solutions.
right.
So, can these structure affect the behavior of proteins?
Of course. I mean, if you talk about the biological perspective of any protein, right? So there is always a post-translational modification. But whenever we make a sensor, we are very much sure about the protein or very much sure about the target what we are going to detect. So I said earlier also, we need to know the biomarker system. If some protein is changing completely, right, very pronouncedly in the cytoplasmic system, or because of the post-translational modification, or because of any reason, we cannot make a sensor for that, right? So that is why you cannot find any sensor for AIDS, right? COVID-19 has also this problem because the target is not fixed. People are somebody is focusing on spike protein, somebody's protein are focusing on another protein. Unless until the target is not fixed, it is highly specific towards a particular disease or a clinical condition, we cannot make a sensor. Now I'm coming to your point. What you said, it is changing within the cytoplasm. In my view, we will not target that protein for any diagnostic based approach. We need to be extremely sure about the target, what we are going to aim for designing any sensor. All right. You understood what I said?
Yes, sir.
Okay.
And sir, how you indicate the performance of any nanobiosensors?
Yeah, performance was basically, it was measured with the standard calibration analysis, what I have shown you. So there could be more things, but that performance is basically the values of limit of detection, the limit of quantification, the dynamic range. The LOL is not that great, but dynamic range and these three parameters and the implication in the real sample, how it is working. So these are the performance parameters. In addition, I said there could be a selectivity coefficient calculation. So this will come in the next phase. First, you need to have a standard calibration. Standard calibration, when you are fixed based on these numerical values, when will obtain. After that, you will go for the selectivity coefficient, the real sample analysis and so on. So these are the parameters what we test for the performance checking of any sensor. All right.
Okay, sir. Yes, sir.
Hello, sir. Hello.
Yeah, please tell me. Yes.
Yeah, sir. Actually, magnetic nanoparticles and quantum dots.
Mm-hmm.
I am an electronic student, so these
Okay.
two terminologies were used in for nanotechnology and
Right.
electronic devices for
Right.
sensing.
Right.
So, would they be also be very effective in this biosensors,
Of course, it will be definitely.
or there
Right.
is any particular different method for detection, like gold
No,
nanoparticles or
like
something like
quantum dots, basically most of the people they are going for optical analysis. Quantum dot and the magnetic nanoparticle, they basically use for the separation. For example, they can separate and detect. So that is basically used for most of the cases. For example, instead a gold nanoparticle, you prepared any particle which is having a magnetic property. Whether that magnetic nanoparticle, the magnetic property of nanoparticle has been utilized for the signal output. That's what is important, right? So it depends actually on what type of the magnetic behavior you are utilizing. I mean, you can simply say that I designed a magnetic nanoparticle, we have loaded that magnetic nanoparticle in the matrix, and the sensor is working well. Whether the magnetic property has been utilized for a signal output, that matters a lot. And with reference to quantum dot, there are so many papers based on the optical sensor, based on the electrochemical sensor. There are so many. Even I saw 2 days back there was one paper from
that?
Italy.
Yeah, sir.
Dr.
Research
Sabina
topic.
might be you have seen for that is on COVID-19 actually. So they have utilized.
Actually,
Hello?
Yeah, sir. Yeah, it's a research topic of quantum dots and other things,
Right.
and people are actively using it
Sure.
for in nowadays.
No, that is absolutely fine. These materials can definitely be utilized for any kind of the. I mean, see, I discuss largely upon the electrochemical sensor, but there are various other sensors. I said also optical sensors, calorimetric sensors, piezoelectric sensors, and so on. There are many things. So various other nanomaterials can be utilized for different other readout systems, right? So that is, of course, these materials cannot be ignored. They can be used, definitely. Yeah. All right.
Okay, so I consider there are no more questions. And if you have any questions, sir has already given the email ID.
Yeah, please write me.
Uh,
No issue.
yeah. So Dr. Chandra, on behalf of Srishti and my team and from Srishti Parivar, I'm really thankful to you for your session and for your time. You have
Yeah,
accepted
thank you.
our
Thank you.
invitation in very short time. That's really great. And we always look forward for your support and guidance for such workshops.
I will be very much happy. Thank you.
Thank you, sir.
Thank you so much.